All right, so we said that during development, myostatin can increase the number of muscle fibers that you have. If you block myostatin, you get more muscle fibers. Or in the adult, it can change transcription and increase or alter mTOR activity. And I haven't shown you that yet, but I'll show you that in a bit. And that's a way that it could increase muscle mass. All right, so the question is, what happens with training? And so this is an experiment where they did animals that did concentric exercise, eccentric exercise, or isometric exercise. And this is 4 days of resistance exercise for these rats. And what you can see is that with the concentric exercise, we decrease myostatin mRNA. It's decreased even further in the eccentric group, and the isometric group looks pretty much like the concentric group. Okay, so what this is potentially saying is that myostatin mRNA is regulated such that when the load is the highest, because the eccentric movement has the highest load, then it goes isometric, then concentric. So when the load is higher, it's suggesting that myostatin mRNA is going down. And that's really interesting, because that would support this idea that if we can decrease myostatin mRNA, we're going to grow our muscle. If we go back to some of the work that's been done using this model of the extreme responders, moderate, and non-responders, myostatin mRNA at baseline starts relatively similar, and it goes down acutely after exercise, and then 16 weeks after training, it goes down with exercise, showing that myostatin mRNA can go down following resistance exercise. But the interesting thing to look at here is that the extreme responders that are growing 15-20% muscle mass over the 12 weeks, they actually have more myostatin than the non-responders, who are, some of them are going down in the amount of muscle mass that they have after 16 weeks. So what this is suggesting is that yes, myostatin goes down with exercise, it goes down acutely and with chronic exercise, but it maybe isn't telling us exactly what's going to happen within the muscle fibers, because here, the ones that aren't going to increase muscle mass, the non-responders, still have the same, if not a greater, decrease in myostatin than the ones who are going to increase their muscle mass extremely. And so what this is potentially saying is that it's not going to predict what's going to, at least the mRNA level of myostatin isn't going to predict what's going to happen. And if you look after a single bout of resistance exercise, half an hour and a half, 3, 6, 18 hours later, and we try and figure out what happens to SMAD2 or SMAD3 phosphorylation, you can see that the SMAD3 phosphorylation doesn't really change across that time. So there's no alteration in SMAD2 or 3 phosphorylation. But again, it's really hard to tell, because if you decrease myostatin mRNA, does that mean that you have less myostatin protein? Or do you have more follistatin or less follistatin? Do you have more of the receptor or less? Do you have more of the ski, snow, and notch proteins? So really, the biggest problem here in the field is that when you try and measure myostatin mRNA, it doesn't tell you anything about what it's doing, because there's 100 different, or not 100, but there's 7 different ways that we've talked about that you can block myostatin between the mRNA, which is way over here, and what it actually is trying to do, which is alter transcription. So what we decided was we needed to figure out a gene that was regulated by myostatin, so that we could identify, okay, that gene is regulated by myostatin. So now we have a target to say, when that gene changes its expression, that gives us a sense as to what the whole pathway is doing. All right, and lucky for us, there's a group out of New Zealand who actually had just discovered this gene, and they called it, we liked what they called it because they called it Mighty. So Mighty, again, is this gene, and is a gene that's got another name as well, but the Mighty gene name is more appropriate for what we're looking at. So if you look and you increase myostatin, what you see is that Mighty expression goes down, and it goes down in a dose-dependent manner, and it plateaus around this 4 to 6 micrograms of myostatin. So what it's telling us is if we add more myostatin, we're going to decrease Mighty gene expression. They did a number of other experiments to say that if they activated the SMAD2/3 proteins, they got a decrease in this. If they constitutively decreased the SMAD2/3 proteins, you actually got an increase. So it really seems like Mighty is a gene that tells us what's happening throughout the whole pathway of myostatin. Because if you add the propeptide of myostatin, Mighty goes down. If you add more myostatin to cells, Mighty goes up. And so it's really giving us an output of this whole pathway. And so now the question is, what would happen if you put Mighty into a muscle? And so the same group put Mighty into a muscle, and they used muscular dystrophy animals. But what you could see is when they added Mighty versus the control, so if they just put in the control vector that held the Mighty piece of DNA, what you got is you got muscle fibers that were, about 200 pixels in size. When you added, instead you got Mighty, you got the Mighty cDNA into those muscle fibers, they went up to about 500 pixels in size. So you had this really significant increase in the average muscle fiber size. And so that's suggesting that Mighty is potentially a downstream target of myostatin that could be involved in regulating muscle size. So then we decided to look at Mighty expression following resistance exercise. And again, going back to the model that we've used in the past, what we then looked at is the change in Mighty mRNA 6 hours after resistance exercise, and that's on the x-axis here. And then we looked at the change in muscle mass from our previous study. We used that study as a guide. And then what we did is we used the 4 different muscles again, and what we could see is that just like mTOR activity, Mighty expression correlated really quite well with the increase in muscle size that happened following resistance exercise. And so what this suggests to us is that Mighty is a good measure of myostatin activity, and it suggests that myostatin could very well be regulated by resistance exercise in a way that could potentially play a role in how big the muscle fiber is going to get. So I did show you, though, that we didn't see any change in SMAD phosphorylation. So then the question became, how is Mighty being activated? And so what we did is we started looking at all these other things in the pathway. So we looked, and follistatin actually goes up. Myostatin actually, after an acute bout, for us, it actually went up at the time point. So it wasn't because we were stopping things at the top. We looked at the receptor, no difference in the receptor. We didn't see any difference in the SMAD2/3. So then we started to look at those transcription factors, SCI, SNO, and Notch. And so SCI and SNO, we didn't see big differences, but active Notch seemed to be something that acutely changed really quickly after resistance exercise. So the active form of Notch is actually, you have Notch as a transmembrane protein, and when it becomes activated, you break off the bottom inside part, and that can go right into the nucleus and transcribe or transduce the fact that there's a change in the extracellular environment. And what you can see is the amount of the active form of Notch is increased for the first 6 hours after resistance exercise, and then by 18 hours it seems to come back down to normal. Okay, so basically what we were showing is that if you cleaved off this Notch, you got this increase in Mighty expression. And so we redid this to see if mTOR was required for it. And so what we did again is we looked to see what happened to active Notch, time course after resistance exercise. So you can see that it's up by 1.5 hours. And it's a little bit, it's still elevated at 3.612, and it's back down to normal by 18, just like we had seen in the past. And that correlated really quite well with this idea that Mighty goes through this increase. It comes down a little bit, and then there's this little kick up at the end that we don't really know what's going on there, but it looks like this increase in Mighty expression after exercise correlates with this increase in Notch cleavage, or this little bit of Notch that goes in and acts as a transcriptional repressor of the SMAD2/3 molecule. And if you look at what happens with rapamycin, rapamycin really doesn't have a dramatic effect on it. The rapamycin dot at 6 hours is in exactly the same place as the control dot, again suggesting that mTOR maybe plays a role at the initial part, but we don't really think it's a dramatic role. So what we're thinking is that Notch cleavage is something that's independent of mTOR and that leads to the activation of Mighty. All right, so this signaling then would go down along parallel to what normal myostatin signaling would be. So Notch again is this transmembrane protein, and you can think about it because the membranes are here for your muscle fibers, and if your muscle fibers are struggling, you're going to get a little bit of movement between the fibers. And so it's possible that that movement produces a shear force, which cleaves the Notch molecule here to make what's called the Notch intracellular domain. And Notch intracellular domain can move straight into the nucleus and bind to the DNA and block SMAD2/3 phosphorylation, resulting in mighty expression. And whether it's mighty that goes directly on to do something important to grow muscle, or it's another target that we don't know of yet, what we're seeing here is that you can locally block myostatin activity using this notch intracellular cleavage. Because now it doesn't matter how much myostatin is circulating, only in the muscle that's exercised are you going to cleave notch. So if I work out my right arm, I'm cleaving notch in my right arm, not my left arm. My left arm's not going to grow, my right arm's going to grow. So now we have a way that we can localize the activity of myostatin, the decrease in myostatin activity, to the muscle that was exercised. All right, so this is a different way of activating. This is a non-traditional way of activating this TGF-beta family member pathway. We're actually inhibiting it. And it's a way that we could potentially be mediating some of the effects of resistance exercise.